Welcome to the series of the Showcasing Discoveries at the Josep Carreras Leukemia Research Institute open lectures of its group leaders. So today, after the Easter break, I have the pleasure to introduce Dr. Ruth Risueño. So she did her PhD in molecular biology at the Autonomous University of Madrid in Spain. In the field of immunology, and then she moved to Canada to perform her postdoctoral training at the McMaster University. And during this period of time, she focused on stem cells and their application in cancer. And then since 2011, Dr. Risueño leads the Leukemia Stem Cells Group at the Josep Carreras Leukemia Research Institute. And her group is focused on the search for therapeutic targets and biomarkers for leukemia. And in addition, since 2015, Dr. Risueño co-founded Leucos Biotech with a company with the aim to clinically develop new drugs for leukemia. So I just wanted to add one comment. So please remember to send your question to the chat. And basically that's it. So please remember to do it. We want to see your questions. So please, Ruth, go ahead with your presentation.
Hello, good afternoon, everybody. Thanks, Viola, for your nice introduction. I'm gonna start talking about what we do in our lab. So first, I'm gonna start explaining a little bit about the asymmetric division, which is the cell division that is found in stem cells. When a stem cell is divided, it can give rise to two daughter cells that are different between them. One can remain as a stem cell and the other one as a progenitor. This type of division allows a stem cell to maintain their pool, but also generate all the progenitors needed in order to maintain all and generate all the mature functional cell types found in the tissues. The rest of the cells in the body divide symmetrically, giving rise to two daughter cells that are the same between them and may or may not be the same as the mother cell. There is a balance between this self-renewal capacity and differentiation, and in reality, this is tightly regulated. In cancer stem cells, you also find this asymmetric division, but this balance favors the self-renewal capacity, so that a lot more cancer stem cells are generated than progenitors. The first tumor where a cancer stem cell population was identified was acute myeloid leukemia, or AML. In AML, there is a subpopulation of leukemic stem cells that can self-renew and differentiate, giving rise to all the leukemic cells found in a leukemic patient. Due to these stem cell properties, AML samples, patient samples, can be transplanted into immunodeficient mice, and after several weeks, we can find a human leukemia inside the bone marrow of those mice. And this human leukemia resembles the one seen in the patient. Again, and due to these stem cell properties, these leukemic stem cells are quiescent, they are resistant to pro-apoptotic stimulus, and they have an increase in MDR activity. The identification of the leukemic stem cells had a therapeutic implication. Conventional therapy targets proliferating cells. Therefore, the majority of the leukemic cells are going to be affected. However, leukemic stem cells, because they are quiescent, they are going to remain unaffected, and they can initiate again the disease. And this is a relapse episode, which is the main clinical challenge that a physician has in clinic. If we develop an anti-leukemic stem cell therapy, we are going to eliminate the leukemic stem cell population. However, the leukemic cells are going to remain alive. It's also true that these cells, they have a short lifespan, so eventually they are going to die and we are going to eliminate the disease. What we aim to find is a combination between conventional therapy and anti-leukemic stem cell therapy that eliminate all the cell types found in the leukemic, both the leukemic stem cell fraction, but also the rest of leukemic cells. So the hematopoietic system, like the majority of the tissue in the body, is structured as a hierarchy where we can find the hematopoietic stem cells on the top. Those cells, they can self-renew, of course, because they are stem cells, but they also give rise to all the progenitors that we are going to see mainly in the bone marrow. And in turn, those progenitors are going to generate all the functional mature blood cells. This process occurs inside the bone marrow, but the last step of the development of T cells and other lymphoid-related cells that happens in the thymus. In AML patients, there is an accumulation of transformed immature myeloid progenitors in the bone marrow, and this accumulation also reduces the normal hematopoiesis. Acute myeloid leukemia is a disease with an unmet need in the sense that the majority of the patients are going to respond initially to the chemotherapy. However, less than 30% of the patients are going to be alive 5 years after diagnosis. Therefore, there is a need to identify new therapeutic approaches for this disease. We go back to this balance between self-renewal and differentiation. We've seen that in hematopoietic stem cells, this is tightly regulated, but as it is in leukemic stem cells. And in leukemic stem cells, this balance favors the self-renewal capacity. If we are able to push this balance toward myeloid differentiation, we are going to eliminate leukemic stem cells because they are going to they are not going to have this self-renewal capacity anymore, and we are going to exhaust this pool. And the aim of our group is finding differentiation inducing therapies as an indirect way of eliminating these leukemic stem cells. We started looking in the literature for terminal myeloid differentiation genes associated to this differentiation process from leukemic stem cell to more mature leukemic cells. And we perform an in silico screening looking for already approved drugs that may induce this differentiation gene signature. We got several hits that we validated in vitro, ex vivo, and then finally in vivo, and we aim to translate those results into clinics. The first hit that we identified was the serotonin receptor inhibitor. We took several AML cell lines and we treat them with serotonin receptor agonists and antagonists, all of them subtype specific. And we saw that only in the case of serotonin receptor 1 antagonist, we were able to induce cytotoxicity of the AML cells. Indeed, this treatment induced also terminal myeloid differentiation, as seen by the expression of differentiation associated markers and morphological changes. The cytotoxicity induced by these serotonin receptor 1 antagonists were dose and time dependent, and indeed all the AML samples that we tested were sensitive to this treatment. This cytotoxicity was even higher in the leukemic stem cell enriched fraction of these primary AML samples. We move to the in vivo model where we injected AML cells into conditioned mice, and once the leukemia was regenerated inside the mice, we treat those mice, and we finally analyze how the leukemia was developed. The treatment with our serotonin receptor 1 antagonist severely reduced the leukemia burden in vivo. Indeed, if we took primary AML samples and we treat them ex vivo overnight for 18 hours, and then we transplanted into immunodeficient mice and we left those mice untreated, we saw a reduction in the engraftment capacity. This engraftment capacity is a readout of the leukemic stem cell functionality remaining upon treatment. When we took those engrafted cells and we transplanted it into a secondary recipient and we left again those mice without treatment, we saw that the majority of the mice whose samples were treated at the very beginning of the experiment were unable to regenerate the disease anymore. And those that were, the engraftment level was really low, which means that the leukemic stem cell functionality of those primary recipients was severely affected, suggesting again that the inhibition of the serotonin receptor 1 impairs heavily the leukemic stem cell fraction. We perform exactly the same experiment, but instead of using AML cells, we use hematopoietic stem cell healthy cells. And we treat them and inject it into a primary recipient. We didn't see any difference in the engraftment capacity of those leukemic stem cells, regardless the treatment. And we didn't see any effect in the engraftment regeneration, not in the frequency of each of the blood subpopulation. When we performed the secondary transplant, we got exactly the same regeneration levels, which means that the normal hematopoiesis was not affected by this treatment. So we were able to prove that the serotonin receptor can be inhibited in preclinical experiments, and this can be done as a therapeutic approach. We wanted to study was the role of the serotonin receptor itself. We started phenotyping primary AML samples for this receptor. We saw that all the AML samples that we tested so far are positive for this receptor, and indeed there is a correlation between the expression of the serotonin receptor 1b and also 1a, but mainly 1b, and the clinical outcome. That is, the highest expression of this receptor, the worse the clinical outcome of the patient. And indeed, this expression was significantly higher than the healthy donors. We wanted to genetic validate the serotonin receptor 1b as a target for AML. We performed knockdown experiment using an shRNA strategy, and we saw that once the receptor was knocked down, there was a reduction in the proliferation rate and also the clonogenic capacity. When we overexpress the wild-type isoform of the serotonin receptor 1b or two constitutive active isoform of this receptor, we saw that the proliferation rate of these cells increased as compared to the parental one, and the clonogenic capacity also. The serotonin receptor 1b is a GPCR coupled to a G-alpha I. When the receptor is activated, the G-alpha I inhibits adenylyl cyclase, and this inhibition reduces the amount of cAMP that is found in the cytoplasm. This reduction in the cAMP goes also with a reduction in the intracellular calcium. When we inhibit this receptor by using those antagonists, we were able to detect an increase in the cAMP intracellular levels and also an increase in the intracellular calcium. Therefore, this signaling pathway was validated. We also wanted to see what's the effect of the natural ligand, the serotonin, on this receptor in AML. However, we've never seen any effect of the treatment with serotonin in AML. Although we are able to see an effect in signaling using antagonists for this specific antagonist for this receptor, which suggests us that the serotonin receptor 1b in a leukemia context is constitutive active, or the signaling basal level is way higher than in other cell types. We also wanted to study whether this receptor was also overexpressed in other blood cancer related to AML, and we analyzed MDS and CMML. In both cases, the expression of the serotonin receptor 1B was overexpressed, and it was significant. And indeed, the treatment with serotonin receptor 1B specific antagonist reduced the cell viability of primary samples from these two diseases. So in a normal situation, on the leukemia cell, the serotonin receptor is actively signaling, and this produce at the end an increase in the activation of P38, AKT, and ERK that eventually give proliferation and cell survival signal. When we inhibit this receptor, the signaling pathway is blocked, and the decrease in those secondary messengers produce an activation of the differentiation pathway and an induction of the cell death, together with the increase in calcium, intracellular calcium. Serotonin receptor 1B antagonists are approved for the use in human. However, those that are approved are not specific for this serotonin receptor subtype. Indeed, they blocked several receptors inside the serotonin receptor family, but also in the dopamine. Therefore, in collaboration with Leucos Biotech, which is a spin-off company from the institute, we started to develop a proprietary small molecule library in order to aiming to find a new chemical entity as a small molecule with a high specificity for serotonin receptor 1B that do not cross the brain-blood barrier, oral, to be oral with a high oral viability, with a optimized pharmacological profile for leukemia, and to be safe. We did several cycles with the medical chemistry program, and at the end we got several lead compounds that we are clinically developing. Indeed, some of them has been already proved in relevant in vivo preclinical models of AML, and all of them they show a very interesting, highly specific anti-leukemic effect. So in this part, what we've described is serotonin receptor 1B as a new therapeutic target for AML and other related neoplasms. And indeed, we've developed new serotonin receptor 1B antagonists with an interesting pharmacological profile. Right now, we are trying to understand better the signaling pathway modulated by this receptor in leukemia, and we are also finishing the IND-enabled study to start clinical trial as soon as possible. Another hit that we obtained from this first screening was the prolactin receptor inhibitor. The first thing we did was study whether this receptor is expressed in AML, and it was because at the beginning it sounded a little bit strange that a prolactin receptor could be expressed on AML. This receptor is expressed on the surface of the majority of the AML samples that we tested, and indeed the expression was significant as compared to the healthy blood cells. Interestingly enough, we've never seen any difference in proliferation in the presence of prolactin or an antagonist of the ligand. Therefore, it seems that the signaling pathway modulated by the prolactin receptor is not involved in proliferation itself. However, when we treat AML cells with this prolactin mutant, we do see a significant reduction in the clonogenic capacity. The prolactin receptor is a monomer that once the ligand is recognized, it forms a dimer and it starts the signaling. There are several isoforms in the nature, but we are studying two, a long and a short. The long is the conventional one where you have all the domains, signaling domain, whether it's short, lacks the intracellular tail. Therefore, their signaling is impaired. So we generated AML cells that express either isoforms in a high and low level, and we saw that the expression of the wild-type isoform induced the proliferation of the cells, whether the expression of the short and then dominant negative isoform reduced the proliferation of the AML cells. Indeed, we've seen that there is a relationship between the expression of the prolactin receptor and the sensitivity to cytarabine, which is the backbone of the AML treatment. The highest the expression of the prolactin receptor, the highest the resistance to the cytarabine. And finally, when we transplanted the AML cells into immunodeficient mice and then we treat them with prolactin, we did see an increase in membrane potential in both genders. So far what we have is that the prolactin receptor seems to be a biomarker for AML and other related neoplasms, and the signaling pathway modulated by this receptor is implicated in survival, mobilization, chronogenicity, and chemotherapy resistance. What we are doing again is trying to identify which signaling pathway is implicated in leukemogenesis and is modulated by this prolactin receptor, and also trying to validate this receptor as a potential therapeutic target for AML. So it's true that at the beginning we wanted to identify ways of pushing the differentiation of leukemic stem cells toward mature phenotype, but also we started being interested in trying to find ways to revert early events in leukemogenesis. The MLL-AF9 fusion protein is one of the few fusion proteins that are able to transform a healthy blood cell into an AML. Taking advantage of this system, we were able to identify which genes were upregulated and downregulated during this transition from a healthy blood cell to a leukemic cell. And we looked for already approved drugs that may revert this early transformation signature. Our hits were histamine receptor 1 antagonists, antihistamine, a group of them. So we started taking several AML cell lines and treat them with histamine receptor specific, subtype specific antagonists. And we saw that only a group of histamine receptor H1 antagonists were able to reduce the cell viability of the AML cells. And indeed, those same antagonists induce a clear terminal differentiation of AML. We saw that the histamine receptor 1 is overexpressed in AML, MDS, and CML, but in this case, it's also expressed in other myeloid healthy subpopulation. We try to find a correlation between the affinity of these antagonists and the expression of the receptor or the cytotoxic effect on AML, and we didn't find any. Indeed, we weren't able to revert the cytotoxicity induced by those antagonists in the presence of histamine. So it was clear that the pharmacological effect of this compound was histamine receptor independent. Based on the literature, we were able to determine that those compounds were cut. They belong to this family, which is cationic amphiphilic drugs, and are a family of compounds that share a specific physicochemical property. Thanks to these properties, those compounds cut can cross cell membranes in a passive way, and they accumulate in lysosomes and mitochondria. So we wanted to see whether that was the case, and indeed it was. Treatment with this CAD increased the number and the size of the lysosome, and indeed those lysosomes were permeabilized. They have a permeabilization of their membrane. So our treatment highly disturbed the lysosomal integrity of the AML cells. Indeed, this affection on the lysosome was also seen as an induction of the autophagy, as seen by an increase in cyto-IgE and an increase in LC3B. Therefore, the autophagy was activated upon treatment. But not only the autophagy, the lysosomes were affected, but also the mitochondria. We were able to detect an increase in the mitochondrial ROS together with a decrease in the membrane potential and the activation of the caspase 3 and 7. And indeed, the cytotoxicity induced by these compounds were not reverted in the presence of a caspase inhibitor. Therefore, the cell death that we were observing was due to autophagy and also apoptosis. Indeed, we were able to propose a model where these CAD compounds that they share these specific physicochemical properties, they affect simultaneously mitochondria lysosomes, and the dual targeting of these organelles induce the activation of the autophagic flux and also apoptosis that end up in the cell death. We were able also to demonstrate that the treatment with these compounds was highly safe in healthy cells, and that was because the transformation process and feature requires an increase in the ROS and also lysosomal vulnerability. Therefore, the basal level of both features is higher in tumor cells than in healthy. And this is the therapeutic window that we are taking advantage of. So it's true that there are histamine receptor 1 antagonists. They are approved for their use in human. They are widely used for allergic. However, the pharmacological profile is not the best one, and of course, it's not optimized for leukemia. Therefore, we wanted to initiate a medical chemistry program to develop new chemical entities that mimic the effect of these CADs in leukemia and had an optimized pharmacological profile for leukemia. We had several hits when we performed this program, and there were 3 new chemical entities that they display exactly the same mechanism of action. We were targeting lysosomes, and the autophagy and apoptosis was activated, and also we were targeting the mitochondria. Indeed, treatment with these 3 hits that are different between them reduce greatly the clonogenic capacity of AML without affecting the clonogenic capacity of the healthy hematopoietic stem cells. We also tested one of the HIT compounds in an in vivo preclinical relevant model of AML, where we transplanted AML cells. We leave the leukemia to be regenerated, and then we treat those mice, and we saw a severely reduction in the leukemic burden in those mice treated. So in this part, what we've seen is that the lysosome play a key role in AML, and indeed, we were able to identify CAT, cationic apophilic drug, as a specific and selective pharmacological agent in AML. What we are trying to do right now is a hit-to-lead optimization of those 3 new chemical entities that we've developed, and also we are trying to identify new druggable lysosome-associated proteins, together with trying to increase our knowledge about the role of lysosomes in leukemia. And finally, a few years ago, dopamine receptor were identified as a new therapeutic target for cancer stem cell in general. And indeed, the inhibition of this receptor was described as a way to induce terminal differentiation of cancer stem cells. This work was done in embryonic stem cells and also in AML and breast cancer. Due to the similarities between AML and another acute leukemia, such as T-cell acute lymphoid leukemia, we decided to study whether the dopamine receptor can be used as a therapeutic target in TALL. So the first thing we did, we took several TALL cell lines and treat them with different dopamine receptor antagonists from the 2 families. And we saw that only the dopamine receptor D4 antagonist had an anti-leukemic effect. However, we've never seen expression of the dopamine receptor on TALL. TALL cells, they don't express none of the 5 dopamine receptors. Therefore, it was quite clear that again, the pharmacological effect that we were seeing with this dopamine receptor antagonist was independent of the receptor they were described to recognize. We performed a wide gene expression profile of non-treated and treated TALL cells, and we identified lipid metabolism associated genes as those heavily regulated upon treatment. And indeed, most of the genes key for the lipid synthesis pathway were then regulated upon treatment. Indeed, when we treated the TALL cells, we were able to see a decrease in the amount of phospholipid found in the cells. Those phospholipids were hydrolyzed, and an increase in the neutral lipid were also detected. Indeed, those phospholipids accumulated in lysosomes, and those lysosomes, they increase inside and also fuse to autophagic vesicles. And at the end, this treatment in TALL activated a lysosome-dependent autophagic mediated cell death. We also took those antagonists and treated primary TAL samples, and we saw in all cases dose response and time response in terms of cytotoxicity. Interestingly, we've never seen any effect of this treatment in healthy blood cells. So we moved to an in vivo mouse model where we took the TALL PDX samples and we injected into immunodeficient mice, and we started treating those mice. So far, with all the PDX TALL samples that we've tested, there is a severe reduction in the engraftment capacity upon the treatment with this antagonist. So as a summary, the TALL also present fragile lysosomes similar to AML, and this disruption of the lipid metabolism has an anti-leukemic effect in the TALL. We are currently trying to understand more about this role of the lipid metabolism in TALL, and also trying to identify lipid metabolism modulators that may be used in this disease. And having said that, the serotonin receptor project was done by Amaya Echave, Antonia Banús, Pepe Carbó, and Liz. All the data presented about the prolactin receptor was generated by Laia. The lysosome team is composed by Pepe Cornet, Pepe Carbó, and Jennifer. We are working in close collaboration with Jordi Esteve and his group. He's the head of the hematology department at clinic. We are performing the medical chemistry in collaboration with the MCS group at CSIC in Barcelona, led by Amadeo Llevaria. And we have to thank all the help from the IGC Biobank, led by Kiko Sole. And if anyone has questions, I'm more than happy to take them.
So thank you very much, Ruth, for such inspiring talk. We have quite a lot of questions, so I'm gonna start. So we have some of these from Pablo Menendez. So first of all, he said that it's a very interesting work and it's highly applicable, right? So basically, the first question that that he has asked basically is, have you treated AML xenograft in vivo with your compounds in combination with ADAC-C and idarubicin?
Yes, we have, and we've seen a synergic effect,
okay,
which is expected because of the mechanism of action that our compounds have.
Mm-hmm. So another question is, have you treated immunodeficient mice and graft with healthy CD34 cells, right, with your drugs? And in this case, is there no myeloablation in vivo?
No, we've done that, and we have never seen any difference between treated and non-treated mice in terms of normal hematopoiesis.
Mm-hmm. And finally, many AMLs relapse with extra bone marrow disease. Do these compounds reduce extra bone marrow infiltration?
And this is something that we don't know, in the sense that the mouse models that we are using, they are not the best for demonstrating this. So we don't know. It's a very interesting question, but we don't have the answer.
So thanks. Also considering that the serotonin and the prolactin receptor are also expressing in other cell types, normal ones, for instance, neuronal or red cells, right? Did you observe or did you expect that these inhibitors of this receptor can cause side effects? And in this case, which type of side effects did you observe or you expect to observe?
So we performed several experiments where we are injecting normal hematopoietic cells into these mice and then treat them, but also we treat the mice directly, wild-type mice, to see whether there is a toxicity associated, and we've never seen any. And what our data suggests is that in cancer cells, this receptor is in an active conformation and is signaling without the effect of a ligand. And therefore, we are inhibiting a receptor that is constitutively active. However, in our body, the amount of receptors, serotonin receptor and prolactin receptor, that are constitutively active are really low. So as we have a hit and run model of pharmacological effect, it's a way to explain why we are not seeing any significant side effect.
Mm-hmm. Okay, so there is one more question missing. So basically, you have claimed that the serotonin and the prolactin receptor are expressed in AMN, right? But is there any kind of a specific correlation between the expression of the HCR1B or PRL with any specific subtype of EML, or is something that is more general?
As far as we know, we've seen the expression in all subtypes. The only correlation that we've seen is with the clinical outcome. So there is like a prognosis value of the expression of those receptors, even though we haven't analyzed a cohort big enough to prove that really in reality. But we've never seen any relationship with a specific subtype.
Okay, so I think that we don't have more questions. So thank you very much, Ruth, for this nice presentation and also the nice answer that you provide. And also thank you very much for all the audience to listen and also to provide such as interesting questions. So see you next time. Bye.
Thanks. Bye.